In the previous video, we learned how molecules like oxygen and CO2 moved across our cell membranes due to simple diffusion. Remember that this was passive movement, didn't require any energy. Passive movement of a solute, in this case the oxygen and carbon dioxide gas, down each of their concentration gradients. So oxygen is higher outside the cell than it is inside the cell, so it moves in, and vice versa for CO2. But recall that all organisms are in an aqueous medium. We're mostly composed of water. So these solutions are based in water, and those water molecules are active. And so now we want to consider osmosis, which is the movement of water. So take a look at this situation where outside the cell we now have sodium ions in the water solution outside the cell. Now remember that those sodium ions, because they're charged, are unable to enter the cell through simple diffusion. Remember, anything large or charged can't move in easily. So these sodium ions are restricted from being able to move. Also, the fact that there are these charged sodium ions outside the cell and not inside the cell means that if we think about the water concentration, it's actually lower outside the cell. And that's because water likes to interact with those sodium ions, so effectively making it a lower water concentration outside the cell. So when the solutes can't move across the membrane, the solvent does. And there we see water moving down its own concentration gradient from higher to lower concentration. Thus, osmosis is the passive movement of water down its concentration gradient. Okay, so a few notes about osmosis. First of all, it's the movement of water from low to high solute concentration. So remember that water is the solvent, so that's what's different here from simple diffusion, is now we're talking about the movement of the solvent. And that's from low to high solute concentration because we always want to talk in terms of the solute concentration of solutions. Think of this as water wanting to dilute the more concentrated side. Now, water isn't a living organism or anything, so it doesn't want to do anything, but that's a good way to think about it. It's always going to want to move to that more concentrated side. And to determine whether or not water is going to enter or leave a cell, we need to consider what's called the tonicity of the solution outside the cell. And tonicity just means what is that solute concentration outside the cell relative to that of the cell itself. Okay, so let's consider this aqueous solution of 12 grams sodium chloride per liter. Now this is an aqueous solution, so let's add some bubbles here to give it that aqueous feel. And say we have a cell immersed in this solution that's 9 grams of sodium chloride per liter inside. Thus the solution outside would be what we call a hypertonic solution, which means higher solute concentration. Hyper, that prefix means higher. So because of that, water will want to move outside the cell in order to dilute the more concentrated side. So in this case, water will always want to move into the hypertonic solution and will leave us with a shriveled up cell. Okay, so now let's consider the opposite situation. We'll put our same cell in a solution that is now lower outside the cell, 5 grams of sodium chloride per liter. And this is what we would call a hypotonic solution. Hypo meaning lower. So in this case, the more concentrated side is inside the cell, so water will move in. And this will serve to expand the cell greatly. In plant cells, that would be expanding up against their cell walls, for instance. And so water will always enter the cell if it's placed in a hypotonic solution. In the last case, say we put our cell in a solution that has the same solute concentration. In this case, water still moves in and out of the cell at the same rate, and that's what happens in an isotonic solution where there's equal solute concentration. So here we see water moving in and moving out, diffusing across that membrane at the same rate. So there's no net gain or loss of water. Okay, so Take a look at these 3 different flasks that each have a cell in a different solute concentration. Now, what I want you to do is pause the video in a moment and ask yourself for each flask, is the cell in an isotonic, a hypertonic, or a hypotonic solution? And then determine whether or not water is going to move into the cell, out of the cell, or if there's no net water movement at all. Okay, so let's consider situation A. The outside solution is higher in concentration than inside the cell, so this would be a hypertonic solution, higher. And in that case, water wants to dilute the more concentrated side, which is outside, so water will leave the cell. Now, in situation B, we have both concentrations the same, thus this is an isotonic solution. And in that case, no net water movement will occur. In other words, water will move in and out at the same rate. In situation C, we have a lower concentration outside the cell, and that's what we call a hypotonic solution. And in this case, water will enter the cell to dilute the more concentrated side.